In today's video, we're going to be taking a look at the nervous and endocrine systems for A-level biology. Now, the first thing that we need to understand is what is a stimulus and a response. So a stimulus is any change in the internal or external environment that has the capability of bringing about a response in an organism. Now, as I mentioned, we have internal and external stimuli. Internal stimuli are generally related to the tissue fluid or blood, and some examples are glucose blood concentration levels, water potential in the blood, carbon dioxide concentration. Some examples of external stimuli are things like temperature, touch, sound, gravity maybe as well. Okay, so these are examples of stimuli. Now, response, so the ability to respond to a stimulus actually acts as a selection pressure. If, for example, we have two organisms, okay, and one of them is able to respond better to sound stimuli, it will have a higher chance, possibly, of being able to survive and pass on its alleles and create offspring, passing on its alleles, right? If we have two organisms, and one of them has the ability to respond to the sound stimulus of a predator running towards it, and it can evade the predator, it will be able to survive. And compared to another organism that is unable to respond to the stimulus of the sound of a predator running towards it, right? That organism would die. And so this organism that is better able to respond to the sound stimulus will have a higher chance of survival and a higher chance of being able to produce offspring, therefore being able to pass on those favorable alleles. So that is how the ability to respond to a stimulus is actually acting as a selection pressure. So the endocrine system is made up of glands and the secretions of those glands called hormones. Now, the three main hormones that we need to know for A-level biology are the antidiuretic hormone, abbreviated as ADH, which is involved in the control of water potential in the blood. We then have insulin, which is involved in the reduction of glucose concentration in the blood. So when the glucose concentration is too high, insulin will be produced and released, which will enable the lowering of the glucose concentration in the blood. Now, this will actually occur by a couple of different methods, which we'll look at here. Number one is glycogenesis can occur. So what this means is glycogen will be formed from glucose molecules. And what this does is it will maintain a steep concentration gradient for glucose between the cell and the tissue fluid and subsequently the blood. So what this means is because in the cell glucose is being converted to glycogen, glucose will diffuse from the tissue fluid into the cell. And that means that glucose from in the blood will diffuse into the tissue fluid, okay? So the insulin is really working to move glucose out of the blood and into the cells, right? We want to move glucose away from the blood. What will also happen is in the adipose tissue, glucose will be converted into fat, and this will be a source of energy later on. On the other hand, glucagon works antagonistically, okay? So it's doing the opposite of insulin. So when the glucose concentration in blood is too low, glucagon will be produced and released. And one of the things that this will do is glucogenesis. So glucogenesis will be where glycerol and amino acids are converted into glucose, okay, which can enter into the blood. Now, these are just a couple of examples of the effects that insulin and glucagon have on reestablishing a normal glucose concentration in blood. Okay, so there are many more different things that do happen, but these are just some examples. Now, glucagon is produced and released by the A cells of the islets of Langerhans in the pancreas. Okay, so the pancreas has a group of cells called islets of Langerhans, and there are two types, A cells and B cells. The A cells are larger and they produce and release glucagon, and the B cells, which are smaller, produce and release the hormone insulin. So where the method of communication in the endocrine system is chemical with the use of hormones, in the nervous system, it is electrical. So the ions will generate nerve impulses. Now, in the nervous system, there are two major divisions. There's the central nervous system, and there is the peripheral nervous system. Now, the central nervous system can be abbreviated as the CNS, and this is the brain and the spinal cord. The peripheral nervous system can be abbreviated as the PNS, and this is composed of pairs of nerves that originate from either the brain or the spinal cord. Now, the peripheral nervous system can be divided into two main groups, into two main nervous systems. The sensory nervous system, which carries nerve impulses towards the central nervous system, and the motor nervous system, which carries nerve impulses away from the central nervous system. Now, the motor nervous system can be further subdivided into two other groups. The somatic nervous system, which carries nerve impulses to skeletal muscles and is under voluntary control, and the autonomic nervous system, which carries nerve impulses to cardiac muscle, glands, and smooth muscle, and is under involuntary control. Now, we know that the sensory nervous system carries nerve impulses towards the central nervous system, and the motor nervous system carries nerve impulses away from the central nervous system towards effectors like muscles or glands. Now, the way to remember which is simply if we understand that the sensory nervous system is afferent and the motor nervous system is efferent. If you think about it, if we have an effervescent tablet and we put it in water, it will dissolve, right? The particles are moving away from that initial tablet. So when I see motor nervous system and I know it's efferent, I know that means, okay, it's going to be carrying whatever it's doing, it's going to be moving away from that initial thing, which is that central nervous system. So we can remember this, motor nervous system will be carrying the nerve impulses away from the central nervous system because it is efferent, and therefore the sensory nervous system, which is afferent, is going to be the opposite, and it's going to be carrying nerve impulses towards the central nervous system. Now, before we move on, let's take a look at some of the main overall differences between the endocrine system and the nervous system. So the first thing that we already took a look at was the mode of communication, right? The endocrine system is chemical with the use of hormones, whereas the nervous system is electrical with the use of ions to generate a nerve impulse. Okay, so that's the first thing, the mode of communication. The second is the mode of transmission. So the endocrine system will release the hormones into the blood stream. Okay, so the glands of the endocrine system are ductless. Okay, so they just directly release the hormone into the tissue fluid, which will then go into the blood stream. Whereas the nervous system will transmit the nerve impulse along an axon, as we will get into later on. The speed of transmission also differs. In the endocrine system, the hormone will have to enter into the bloodstream and then it will have to go and reach the target cell. Whereas the nervous system has a very rapid mode of transmission, right? The speed of transmission is very quickly because these nerve impulses are being transmitted from neuron to neuron extremely quickly, right? We can have a very fast reaction time. That's the neurons all being involved. We also have the destination. So with the hormones in the bloodstream, they're going to be going all around the body, despite actually only being for a specific cell or a group of cells. Those hormones are going to be traveling all throughout the body within the bloodstream, whereas the nervous system is very specific. It will only go to a specific effector within the body. It's not going to be going everywhere. The nerve impulse isn't going to be going everywhere in the body before reaching where it needs to go. We also then have the response. So the endocrine system, with the use of hormones, will generally stimulate a response that is much slower. However, it is normally going to be permanent. Okay, let's say, for example, in puberty, the release of hormones will stimulate growth, and that growth is irreversible. On the other hand, the nervous system has a response that is very short-lived, and it's also irreversible, right? If the nervous system tells our hand to close, it's going to be short-lived, but it's also reversible. We can extend our hand again. And the duration of the effect. Okay, as we just said, the duration of the endocrine system with the hormones will be irreversible, it will be long-lived, whereas the duration of the effect of the nerve impulses in the nervous system will be very short-lived. So now let's take a look at the difference between a sensory receptor and a sensory neuron. Now, a sensory receptor is simply a cell, as we will look at later on in the case of a chemoreceptor in the taste buds. It's simply just a cell that can detect a stimulus. On the other hand, a sensory neuron transmits a nerve impulse towards the central nervous system. Okay, so sensory receptors are cells that detect a stimulus. Sensory neurons will transmit a nerve impulse towards the central nervous system. And we can see here, this is the structure of a sensory neuron. It's a lot different from the structure of a basic sensory receptor. Now, motor or effector neurons, as we discussed here, are neurons that are going to carry away a nerve impulse from the central nervous system towards an effector, like a muscle or a gland, right? Skeletal muscle in the somatic nervous system, which is under voluntary control, and then a gland in the autonomic nervous system, which is under involuntary control. So now let's take a look at the different neurons that we have. The first is going to be the sensory neuron. We can see here that we have a dendron that subdivides into these smaller fibers called dendrites. And this is where a nerve impulse will be transmitted to a sensory neuron. Now, as we move along the sensory neuron, we have this axon. We then have a cell body about halfway between the terminal axon and the dendron, right, and the dendrites. And then at the end, we have the terminal axon. And this is where the nerve impulses will be transmitted to another neuron. So this is the direction of an impulse. It is coming into the dendrites, traveling along the axon before being released from the terminal axon to other neurons. So now let's take a look at the structure of a myelinated axon. So a myelinated axon has Schwann cells that wrap around the axon. And the Schwann cells are very rich in the lipid called myelin. And what this does is it will actually insulate, so electrically insulate the axon. So nerve impulses will not be able to be transmitted in these regions where the myelin sheath is, right, where those Schwann cells are. Now, between each myelin sheath, a little space is about 2 to 3 micrometers long, and these are actually called nodes of Ranvier. And this is where the nerve impulses will be transmitted along as they travel along the myelinated axon from the dendrites to the terminal axon. Now, the process in which they are traveling from node of Ranvier to node of Ranvier is called saltatory conduction. And so because of this, because the nerve impulse is jumping from node of Ranvier to node of Ranvier, instead of going across each point of the axon membrane, the nerve impulse is transmitted much faster along a myelinated axon than an unmyelinated axon. Now, let's take a look at the motor neuron. So the motor neuron has the cell body attached to the dendrons and then dendrites. So the nerve impulses will come in through the dendrites, through the dendrons, then they will go through the long axon and then be released out of the axon terminal. They will be passed along out of the axon terminal. In the intermediate neuron, we have all of these dendrons and dendrites that extend from the cell body. Actually, this here is our axon. So all of these dendrons and dendrites are bringing nerve impulses in to the intermediate neuron. And then we have our one axon that will be responsible for passing on the nerve impulse out of the intermediate neuron. So the sensory neurons will transmit a nerve impulse from a receptor to the central nervous system and connect directly to a motor neuron, which will then transmit the nerve impulse to an effector, as we discussed before, or the sensory neuron can transmit the nerve impulse to the central nervous system and then connect to an intermediate neuron, which will then receive the nerve impulse and pass it on to the motor neuron, which will then take the nerve impulse to the effector. Okay, so this is how the sensory motor and intermediate neurons can work with one another. Now, as we see here in the cell body of each of the neurons, there are these little points, and they are the points that represent the endoplasmic reticulum of each neuron. Now, the endoplasmic reticulum of these neurons forms what is called Nissl's granules, and the endoplasmic reticulum is responsible in the synthesis of proteins as well as neurotransmitters, which we will get into a little bit later on in the video on the cholinergic synapse. So now let's take a look at sensory receptors using the example of taste buds. So each taste bud has between 50 to 100 chemoreceptor cells. Now, a chemoreceptor is a cell that can detect chemical stimuli. Now, the chemoreceptors in taste buds are thought to be able to detect the chemicals that are responsible for producing the 5 following tastes: sweet, sour, savory, salty, and bitter. Now, what they will do is they will convert the chemical energy into electrical energy, right? We discussed before that the nervous system, the mode of transmission, the mode of communication of the nervous system is electrical, right? With ions and nerve impulses. So these chemoreceptors will convert the chemical energy into electrical energy. And in doing so, they are actually known as transducers, right? This is called transduction. The conversion of energy from one form to another is transduction, and because chemoreceptors do this, they are called transducers. So let's take a look at how the chemoreceptor cells will react to, let's say, a salty taste. So there will be the sodium ions present in the tongue, right, on the taste buds, and they will enter the chemoreceptor cells via the microvilli, via, more specifically, the sodium ion channels. Now, when they do this, they will depolarize the membrane of the chemoreceptor cell. And this depolarization in a chemoreceptor or in a receptor cell is called the receptor potential. Now, what the receptor potential will result in is that the calcium ion channels will open so that calcium ions will enter the chemoreceptor cell. Now, within the chemoreceptor cell are vesicles that contain neurotransmitters. When those calcium ions enter due to the receptor potential, those neurotransmitters will go and fuse with the cell surface membrane of the chemoreceptor and release the neurotransmitters via exocytosis into the synaptic cleft that chemoreceptor has with a sensory neuron. Now, the greater that the receptor potential is, so the more chemical, let's say the more sodium ions that are entering into the chemoreceptor, the greater the receptor potential will be, so the more calcium ions will enter, so the more neurotransmitter will be released into the synaptic cleft. Now, as we will see in the cholinergic synapse video, this neurotransmitter, if there is enough neurotransmitter, a generator potential will be formed in the sensory neuron, right? So this means that the threshold value will be reached, and that means that an action potential will be achieved, and we will sense, right? We will sense that chemical that triggered that initially triggered the sensory receptor cell, the chemoreceptor. If there is not enough neurotransmitter being released, then a generator potential in the sensory neuron will not be released, and so no action potential will occur, and so we will actually not even sense, we will not detect that chemical that touched the chemoreceptor. Now let's take a quick look at the difference between negative and positive feedback. So negative feedback is when the results of a sequence of events stops that specific sequence from occurring anymore. Positive feedback, on the other hand, is where the result of a specific sequence of events will ensure that that sequence of events continues working. Okay, so negative feedback is when the result will stop the process. Positive feedback is when the result will make sure that that process continues working. So that was the video on nervous and endocrine systems for A-level biology. If you found it interesting, take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thank you very much for watching, and I hope you have a great day. Bye-bye.